We'll continue our discussion of DNA replication with talking about the replication of the telomere sequences, that is, replication at the ends of the chromosomes. Now here you see a schematic of a chromosome, piece of double-stranded DNA with 3 origins of replication. You have 3 replication bubbles forming. When these bubbles collide, that's fine. That means you've completed that section. And since you have the leading strand here, there'll be no problem synthesizing this from 5' to 3' right to the very end of this double-stranded piece of DNA to produce a blunt end. But on the lagging strand right here, you have to jump ahead, synthesize back, jump ahead, synthesize back, and now you can't jump ahead enough to synthesize back all the way to the previous thing. So just to erase this last drawing, how are you going to fill in this region? Because we cannot jump ahead to synthesize back. And here in the bottom, this point is made in a little larger scale. The solution to this problem is a special enzyme, a protein RNA complex called the telomerase. Telomerase is depicted in the protein part is in green, and its RNA component is depicted in blue. The problem that telomerase is tackling is filling in this overhang region where the lagging strand cannot fill in. It does that by actually making the template strand a little longer. This molecule of RNA that telomerase acts as a template for additional sequence at the telomere ends, putting down extra copies of the telomere repeats. I'll say a bit about that in a second, so that you're extending the other chromosome. Now, other strand, now you can put in an RNA primer and fill this end in with the Okazaki fragment, getting it to at least the distance required for the original chromosome end. Now the telomere repeat sequence, these fragments that are repeated over and over again, telomeres actually have thousands of copies of 6 base pairs. It's a hexanucleotide. The sequence is different in different organisms, but they function similarly. In humans, the sequence is TTAGGG. And these thousands of copies of the telomere repeat acts as a molecular clock. Telomerase, the enzyme, is only active in certain cells. They're active in embryonic cells early in development when a human is just forming. They are active in stem cells, which have this infinite renewal capability. And they are inappropriately activated in cancer cells. That's one of the adaptations cancer cells have made to achieve immortality. In the rest of cells, meaning most of our adult cells, our chromosomes are not getting filled in each time, and they're actually getting shorter each cell division. This shortening of telomeres is what acts as a clock. Eventually, the telomeres get so short that a cell is no longer able to divide. These cells are called senescent, and they won't necessarily die, but they can no longer divide. They are just present. You may have heard that you do not make new neurons any longer. These are an example of cells that are senescent. This slide shows some of the details of the RNA template. In this particular organism, the hexanucleotide repeat is a little different from humans, TTGGGG, and we're going to add that over and over again. Now, one thing to consider is how frequently would you expect a repeat to occur? How often would you expect a hexanucleotide to repeat? Since you can have 4 bases at any position, you can have 4 bases at position 1, 4 bases at position 2, 4 bases at position 3. For a hexamer, that would be 4 to the 6th power, or you would expect any particular hexamer to occur once every 4096 bases. Obviously here it's occurring every 6 bases, so that's statistically highly improbable if it were due to chance. But generally it's something you need to know about, is to calculate how frequently you would expect to observe any particular random sequence. And here's another YouTube video that shows the action of telomerase working, which is, if there's any confusion, I recommend that. It's very helpful. Now it turns out that not all organisms have double-stranded linear DNA chromosomes like humans do. There are many other types of genomes. So while we've been talking about DNA replication, the more generic concept would be genome replication. And most of these other genomes are either bacterial or even more so viral. Viruses have just about any type of genome, nucleic acid-based genome imaginable, including some that pose real problems for biological theory. Either genomes made up of either single-stranded RNA or double-stranded RNA. This challenged the assumption that DNA is the ultimate repository of all genetic information, and also there was no known method of replicating RNA in nature. So we'll explore a little bit more about viruses. Viruses are very simple organisms. Some can have as few as a handful of proteins. They don't have all the machinery needed to replicate their genome and put into effect the processes which are needed for replication of the organism and viral life, if you want to call it. The way they do that is by hijacking the machinery of normal cells. In this case, you have a virus with a double-stranded DNA genome. The virus infects most often a bacterial cell, but also could be a eukaryotic cell. It introduces its DNA into the cell and tricks the host machinery into using its proteins to make RNA from the DNA to make proteins, and also to copy its DNA to be packaged inside of the viral proteins. And eventually the virus will cause the host cell to lyse, expelling thousands of copies of virus particles back into the environment. But still we have a problem for viruses that have RNA genomes. And the answer comes from the discovery of a Nobel Prize winning enzyme, reverse transcriptase. And reverse transcriptase is capable of taking an RNA viral genome and making a DNA copy from the RNA molecule. This was discovered by David Baltimore and Howard Temin in the late 60s and early 70s. Once you've made a DNA copy of the RNA genome, you can degrade the RNA and you can create a second DNA strand, making a double-stranded DNA genome, which can then insert itself into the host DNA. This is just the red portion. Now it's possible to make RNA copies of this section, these being both the information to make the proteins needed for new viral particles and also packaged as the genome of for more viruses. But the real revolution came conceptually with the realization that reverse transcriptase violated the central dogma of biology, which is that DNA makes RNA makes protein. Now you had an enzyme which could take RNA and convert it into DNA. This capability is also incredibly useful for biotechnology applications, and we'll encounter it many times, particularly in the third part of the class, when you might want to make copies of all the RNAs that a cell produces and convert them into DNA to have a library of expressed genes. Now, I'd like to leave you with one conundrum that we'll discuss in class, and maybe you can think about how this occurs. Let's talk about the common bacteria E. coli and the replication of its genome. So E. coli, I can tell you, has a single replication of origin. It has a circular double-stranded DNA, and so it forms a replication bubble. It has 2 replication forks. It starts replicating, and we know very, very accurately the speed at which it can replicate DNA, and we can tell you that it takes 90 minutes to complete the synthesis of 2 strands of DNA to create 2 new double-stranded copies of the genome for 2 daughter cells. However, E. coli replicates every 40 minutes. So how can this be? How can the cells divide every 40 minutes when it takes 90 minutes to copy the genome? Think about that for in-class discussion. Thanks.